Hello and welcome to the final seminar series in this third series of webinars of the International Union of Immunological Societies. I am Fernando Martinez Estrada, your moderator for today, and I am a specialist in macrophage biology. I am a lecturer in innate immunity in the University of Surrey in the UK. Today's webinar will be presented by Professor Kate Schroder of the topic of inflammasome signaling from fundamental biology to neotherapeutics. Professor Schroder has the Inflammasome Laboratory and is the director of the Center for Inflammation and Disease Research at the Institute for Molecular Biosciences, IMB, in the University of Queensland, under the National Prestigious Health Medical Research Council Leadership Fellowship. Kate's contribution to the field of inflammasome biology has been tremendous, and I invite you to visit her website in the University of Queensland to see one of the most impressive collections of inflammasome and macrophage activation manuscripts, and a snapshot of the development of this fascinating topic of inflammasome since the very early days of inflammasome research. So one final small note before passing the stage to Kate, to say that to ask you to write all your questions in the Q&A section, that we will be taking those from there. So a very warm welcome to Professor Schroder. The stage is yours.
Thank you so much, Fernando, for that very kind introduction. It is really my enormous pleasure to give this final lecture for the IUIS this year, and to share with you some of the work that we have been doing in the area of inflammasome biology, this really interesting signaling pathway in innate immunity. I'm hoping someone will let me know if my screen is not loading, but without hearing anything otherwise, I'll continue. So I'd just like to start by acknowledging the traditional owners of the land that I'm broadcasting from today. Today I'm broadcasting from the St. Lucia Peninsula in Australia, and this land has always been a space for teaching, learning, research and collaboration for tens of thousands of years before it was established as a university campus as it is today. So I'd like to acknowledge the Turrbal and Yagara people as the traditional owners and the custodians of these lands, and pay my respects to their ancestors and their descendants. So today I'm going to be talking about innate immune system signaling, and really in innate immunity we talk about danger sensing. So danger being anything that's posing a danger to the organism, whether it will be microbes, things like gram-negative bacteria, for example, or what we call endogenous danger signals. These are signals that something has gone wrong in tissue. An example of this would be, say, extracellular ATP, as ATP is supposed to be inside cells instead of out of cells, and therefore extracellular ATP signals tissue damage. The innate immune system recognizes danger through these suites of receptor families, of which the probably most famous are the toll-like receptors. These were the first to be discovered, but there are several other pathways and families that provide surveillance of both the extracellular fluid, internal compartments, and also the cytosol. And it's these cytosolic centers today that I'll be talking about, those that form a molecular complex called the inflammasome. And the sorts of things they recognize include microbial ligands like lipopolysaccharide from gram-negative bacteria, or bacterial toxins like nigericin, or these endogenous danger signals such as ATP when it's sent extracellularly. And I'll be talking primarily today about macrophages, a key innate immune cell, but we work on various innate immune cells, including macrophages, but also dendritic cells and neutrophils, all of which express these signaling pathways and use them to great effect to sense danger. So I'll be talking today about the inflammasomes, and by way of introduction, the inflammasomes are molecular complexes that allow for the activation of inflammatory caspases, and in doing so drive inflammation and cell death programs. So there are many different kinds of inflammasomes. These include AIM2, pyrin, CARD8, and then several inflammasomes that are nucleated by members of the nod-like receptor family, that is NLRP3, NLRP6, NiPE and NLRP4, which collaborate to form an inflammasome, and an area of great excitement at the moment, NLRP1. So I'll be talking today about one particular inflammasome, and that is NLRP3, which is probably the most famous of all of the inflammasomes and perhaps the most problematic in human disease. And this is because NLRP3 is a sensor for cell stress. I always think of NLRP3 as a sensor for any kind of cellular unhappiness, basically anything that indicates that a cell or a tissue is dysfunctional, be it lysosomal damage or mitochondrial dysfunction or membrane damage. These things all activate this NLRP3 protein, which then nucleates the formation of this inflammasome complex composed of NLRP3, shown here in red, ASC, the inflammasome adapter, which is shown here in blue, and this then allows for the recruitment and activation of the inflammatory caspase 1. So the pathway works something like this. Microbes and also endogenous danger signals, things like alarmins like ATP, protein aggregates and misfolded proteins, things like alpha-synuclein and amyloid beta, as well as crystals, things like MSU crystals, gout, cholesterol crystals in atherosclerosis. These all trigger the activation of, in this case, the NLRP3 inflammasome, forming this sort of wheel structure here for NLRP3, which then engages the inflammasome adapter ASC, which forms this fibril shown here, which then allows for the recruitment and activation of caspase 1 and the signaling by caspase 1 through proteolytic programs that include the cleavage and maturation of interesting cytokines, IL-1 family cytokines, pro-interleukin-1 beta and pro-interleukin-18. These are really unusual cytokines because they're expressed by cells as an inactive precursor protein in the cytosol. And this precursor protein sits there waiting for the inflammasome pathway to be engaged, which allows both for its maturation and its release through these pores that are formed by a protein called gasdermin D. So maturation and release of these proteins, of these cytokines, really triggers inflammation, which hopefully serves to inhibit whatever the provoking stimulus is. But however, this can continue to fire, leading to disease. The gasdermin D, this interesting pore-forming protein, is cleaved by caspase 1 and other inflammatory caspases, and this then triggers the formation of gasdermin D pores in the plasma membrane and other membranes, leading to not only the secretion of cytokines, but also this lytic form of cell death that we call pyroptosis. So pyro meaning fire, this is a fiery form of cell death that drives inflammation and provokes innate immune system activation. So these pathways are intended to resolve whatever the provoking stimulus is, but of course, if they're left unchecked, this can cause disease. And this is the reason that inflammasomes are of such clinical and industry interest present, is that inflammasomes drive a really wide variety of human diseases, and you maybe would not think that one pathway would connect such disparate diseases affecting such different organ systems and with very different kinds of pathologies. So in particular, the NLRP3 inflammasome causes several of these diseases, and this includes hereditary fever syndromes that are caused by gain-of-function NLRP3 mutations, various respiratory diseases, so silicosis, asbestosis, and steroid-resistant asthma, various neuroinflammatory diseases, and my lab has particular interest in Alzheimer's and Parkinson's disease. Also metabolic diseases, and here my lab has a keen interest in non-alcoholic steatohepatitis. But really there's a wide array of diseases that are driven by inflammasomes. So this, of course, makes us very interested in how inflammasomes signal, because if we can understand the signaling pathways of of inflammasome-mediated inflammation, we may be able to design drugs to block this pathological inflammation and hopefully reverse or prevent disease. So how might we target inflammasomes in disease? Well, there are already pathway antagonists in the clinic, and these are the anti-IL-1 drugs. These have been around for a while, at least some of them have. So these are anakinra, canakinumab, and rilonacept, and these are biologics that act by either targeting IL-1 or its receptor and blocking the signaling downstream of IL-1 beta. So these are really revolutionary drugs for many diseases, particularly the hereditary fever syndromes and for TNF-resistant arthritis. However, they do have some drawbacks. They're biologics, so they don't penetrate the CNS for neuroinflammatory disease. They're delivered by injection, which patients, of course, don't like. And they only block IL-1. They don't block other functions of caspase-1. They don't block gasdermin D-mediated cell death, and they don't block IL-18. So there's been a lot of interest in targeting inflammasomes further up the pathway with the idea that we may be able to block all of the effects of inflammasome signaling and not just IL-1. So there's been an enormous amount of interest recently in the idea of targeting gasdermin D, with the idea that if you could block lytic cell death, you would also block the release of IL-1 beta and IL-18 cytokines. Although you probably wouldn't prevent cell death entirely, you may push the cell into a less inflammatory mode of cell death like apoptosis. It's actually very hard to stop a cell that wants to die from dying because all the cell death pathways are interconnected. But the idea here is that you could tune down inflammation in disease, which could be very beneficial. And certainly work with gasdermin D knockout mice suggests that this could be a very interesting target. There's also been a lot of interest over the years, actually even predating the discovery of the inflammasome, in the idea of inhibiting caspases. So caspase-1 is the inflammatory caspase that's activated by many inflammasomes. Caspase-4/5 is the other inflammatory caspases that are targeted by inflammasomes. And these are related, of course, to the apoptotic caspases, but they are distinct in structure and function. So I'll touch a bit on caspase inhibitors in my talk today. And more recently, there's been a lot of interest in the idea of stopping signaling by the inflammasome sensor molecule, so in this case, NLRP3, with the idea that if you can block activation of the sensor or signaling by the sensor, you could actually stop all of these downstream processes. And that's really what I'm going to talk about today is the advent of inflammasome-specific inhibitors as typified by NLRP3 inhibitors, which really gives us confidence that this might be a viable approach in future. You know, hopefully we will have a world in some years' time where we have drugs that block every single inflammasome that mediates disease and does so specifically. That would be really the ideal goal. So I want to talk about two projects today from the perspective of how their basic biology informs drug design and how we can target inflammasomes using what we know about the molecular mechanisms of signaling. So I'm going to talk about two candidate inflammasomes drug targets today, and that's caspase-1 and NLRP3. And I'll start with caspase-1 before moving higher up the pathway to NLRP3. So caspase-1 is central to just about all inflammasome pathways. It is central to all inflammasome pathways. It's obligatorily required for signaling by most inflammasomes, whereas the caspase-4/5 non-canonical inflammasome also activates caspase-1, and this is required for cytokine production, but not cell death. So caspase-1 is really central to the pathway of all inflammasomes, mediating gasdermin D processing, IL-1 beta processing, IL-18 processing, inflammation, which often ends up in fibrosis and in disease. So if we could target caspase-1 with an inhibitor, we would actually have essentially a pan-inflammasome inhibitor to block pathology mediated by all of these different human inflammasomes. So why might we want to do that? As I sort of will allude to in the second part of my talk, we are only now just getting specific and potent NLRP3 inflammasome inhibitors, and NLRP3 is really leading the field here, as we do not yet have potent and specific inhibitors for other inflammasomes. So in the short term, caspase-1 inhibitors would provide a mechanism for blocking signaling by inflammasomes other than NLRP3 in disease. For example, blocking NLRP1 signaling in skin inflammation, or NLRC4 in gut inflammation. So that's probably the short-term potential of caspase-1 inhibitors, but the broader potential that I would see for caspase-1 inhibitors in the long term is to block disease in conditions where multiple inflammasomes collaborate to drive pathogenic inflammation. For example, in chronic liver disease, NLRP3 and AIM2 both collaborate to drive inflammation. Likewise, in Alzheimer's, there are reports to suggest that it's not just NLRP3, but also other inflammasomes that drive the pathology of neurodegeneration. So, pan-caspase and caspase-1 specific inhibitors have been under clinical development for decades, but have failed in clinical trials. Today, I'd like to propose that this is because we didn't understand the fundamental biology of caspase-1 activation by inflammasomes. And in revealing that biology, I hope to convince you that alternative mechanisms of action of inhibitors may have better therapeutic benefits. So, caspase-1, how is caspase-1 activated by inflammasomes? Well, actually, when we started this project, we realized that as with many fields that move very quickly, there were some base assumptions made about how caspases were activated by inflammasomes early on in the literature that were never actually really tested. So we thought we'd go back and do those sort of really fundamental experiments to test precisely how caspase-1 is activated. So there were really two major models put forward over the years. One, that caspase-1 would be required to dimerize on the inflammasome to gain activity. And more recently, people have proposed that caspase-1 would need to fibrillate to form a long molecular chain in order to gain activity. And so we thought we'd test this model using a pretty reductionist system. And this is a system in which we remove the card domain, the caspase activation and recruitment domain that allows caspase-1 to bind to inflammasomes. And we replace this with a DMRB domain that allows us to control dimerization with this AP drug. So here we expressed in cells wild-type caspase-1 or a catalytic mutant of caspase-1, this C284A mutation, in which the catalytic mutant, the catalytic cysteine of caspase-1 is mutated, rendering the protease dead. So we express these constructs in HEKs, and they have no activity until we add the dimerizer. And when we add the AP drug to wild-type caspase-1, we now see beautiful activity here read out by a fluorogenic substrate. So this suggested to us that caspase-1 dimerization is sufficient for activity and also necessary for activity. So dimerization alone allows for the activation of caspase-1. And when we remove the card domain, which is actually the domain that fibrillates caspase-1, we still see activity. So this suggests that card-mediated fibrillation of caspase-1 is not required for activity. At least it's not absolutely required for caspase-1 activity. It may still have some effect in vivo in cells. Another thing we were very interested in is caspase-1 self-cleavage. So anyone who's done an inflammasome assay probably has a blot like this. Here we've taken primary bone marrow macrophages, and Dave Boucher, the author of this paper, has stimulated the cells with nigericin to activate NLRP3. And then very quickly, within the 15 to 30 minute time point, caspase-1 very quickly cleaves itself. And if we blot for caspase-1 using an antibody against the large subunit, we see here this free P20 subunit, which is the large subunit of the protein, and also the free P10 small subunit of the protease. So this tells us that both of these linkers, the CAR domain linker and the inter-domain linker, are very rapidly cleaved within caspase-1 within sort of 15 to 30 minutes after stimulation with an inflammasome activator. So the presumption in the field for many years has been that caspase-1 becomes activated on the inflammasome. Caspase-1 then cleaves both linkers to generate this P20-P10 species that we can see on Western blot, and that this P20-P10 species, which is a heterotetrama, based on our dimerization studies, that this P20-P10 heterotetrama zooms around the cytosol and cleaves substrates like interleukin-1 beta. Now, I won't go into all the data today to give you the backstory on why we were a bit skeptical about this model, but basically we used an activity probe to pull down active species of caspase-1, and to our enormous surprise, we could never pull down this P20-P10 species. So this led us to develop an alternative model, which at the time was extremely heretical, and that is that based on our dimerization studies, we suggested that the inflammasome, the major function of the inflammasome is to cluster monomers. Monomers will then dimerize to form this P246 active dimer, and we call this basally active because the only thing it can do is cleave itself, and it does so at the interdomain linker, this linker between the large and the small subunits. So then this generates a species called P33-P10, which has full activity and can cleave substrate. And then after it does so, it cleaves itself a second time, this time at the CAR domain linker, to generate the P20-P10 species that we see on Western blot, and we proposed that this was an inactive form of caspase-1. So again, I don't have time to show you all the data, and you're very welcome to look up the paper if you're interested. But this was a really key piece of data that Dave generated, and that was when he expressed wild-type caspase-1 versus a mutant of caspase-1 in which this CDL cleaved linker is rendered uncleavable. So it locks caspase-1 in this P33-P10 state, and it cannot cleave itself to deactivate. When we express this CDL mutant, we see really beautiful high activity of caspase-1, much higher than wild-type caspase-1. And importantly, while caspase-1, wild-type caspase-1 in red, the activity declines over time, the activity of this CDL mutant is extremely high and it's stable over time. So suggesting that locking caspase-1 in this P33-P10 state boosts its activity and prevents deactivation over time. And we have the catalytic mutant in here as a control. So this was a really surprising finding for us and for others, because many of us, including in my lab, have expressed caspase-1 in a recombinant form as a P20-P10 heterotetrama and found beautiful activity from this recombinant protein. And actually, here's the crystal structure of caspase-1 shown here as the heterotetrama with the two active sites labeled here in blue. So this is really puzzling, because why would recombinant caspase-1 P20-P10 be active and cleave substrate, while cellular caspase-1 P20-P10 does not? So we spent a long time scratching our heads about this, but essentially, to cut a long story short, the thing that's really different between recombinant systems and cellular systems of caspase activation is, of course, that in cells, you need the inflammasome to activate caspase-1. Whereas in recombinant systems, you're expressing caspase-1 to a high level. The high concentration of caspase-1 allows it to dimerize spontaneously without the need for inflammasome, and so triggers the activity of this protease. So let's consider now our model in the context of the inflammasome, and here I'm adding in the card domains of the inflammasome, usually the card domains of ASC, but it can directly of an inflammasome sensor. So when we're considering caspase-1 in the context of the inflammasome now, what you'll notice is that CDL cleavage actually cleaves off this card that allows caspase-1 to bind to the inflammasome. And so now you would expect this P20, P10 to be ejected from the complex. Now, why is that important? This is actually really important because caspase-1 dimers are inherently unstable. The KD, the dissociation constant of caspase-1 dimers, is around 100 micromolar, and the cellular concentrations of caspase-1 are around 10 nanomolar. So that means that at the concentration that's endogenously present in cells, you'd expect caspase-1 to be in a monomeric form and not in a dimeric form. And so you'd expect caspase-1 to only be a dimer when it's tethered to a complex that actually promotes the dimerization of that complex and keeps that dimer stable. So what we did is we then went about trying to test that idea. Again, we used our dimerization system, and here we used a form of caspase-1 that had the DMRB domain instead of the CARD domain. And we also used a catalytic mutant so that it wouldn't cleave itself. And we've replaced the CDL cleavage site actually with a thrombin cleavage site. So that now allows us to control both dimerization of caspase-1 in cells and also its cleavage in trans in a very precise manner. So when we do this, we express this construct in cells. We then added the dimerizer drug to induce the dimerization of caspase-1, and you can see here the P46 dimers that we can capture with a crosslinker. And if we then added AP drug to dimerize caspase-1 and then added in thrombin to cleave the CDL before we add the crosslinker, we can no longer capture dimers. So that indicates that CDL cleavage really does destabilize the complex if it cleaves the protease domain away from the domain that's mediating dimerization. So this has several implications for caspase-1 inhibition, and probably our coolest finding, just from a basic science point of view, is that the whole inflammasome actually functions like a hollow enzyme. So caspase-1 is only active on the inflammasome, and this hollow enzyme has an inbuilt timer mechanism that actually terminates caspase-1 signaling. I teach signal transduction to undergraduates, and I always tell them that signal transduction pathways always have an inbuilt off switch. Well, we think this is the off switch for inflammasome signaling that allows caspase-1 to be deactivated in a timely manner. But when you think about this model, it actually starts to explain some issues with previous active site inhibitors in clinical trials. These inhibitors had poor in vivo efficacy as compared to in recombinant systems. And they also had a high degree of off-target effects, which caused toxicity. And we think this is because they can only bind to active caspase-1. So these are active site inhibitors that are peptidome-mimetics usually. And you actually need caspase-1 to cleave itself at the interdomain linker in order to open up that substrate binding pocket to allow these inhibitors to bind. So with the knowledge now that caspase-1 activity is actually transient and caspase-1 actually very rapidly turns over its own activity, this actually gives a very small window with which these active site inhibitors can work, and so would limit its efficacy in vivo. Also, I haven't talked about this much today, but the ASC spec is a huge structure. It's about a micron wide, and it's quite a dense structure of these ASC filaments. And actually, you're asking a caspase-1 inhibitor to not only penetrate the cell, but also penetrate the ASC spec to inhibit the caspase bound to the ASC spec. And as I said, you'd need to block the activity of caspase-1 before it naturally deactivates to have any in vivo efficacy. So I would suggest that preventing caspase-1 activation may actually be much more effective than inhibiting its protease activity. And I'll leave that part of the project there, but I wanted to give a quick summary of that, as well as introduce the concepts you'll need for the second part of my talk, using the little video that we made as we published this paper in order to explain the signaling pathway as we understood it. This is on YouTube if anyone wants to use it for teaching. So this is a movie told from the perspective of the macrophage cytosol. And in macrophages, NLRP3 is usually held in an inactive state, in this sort of closed state, in which the oligomerization domain, shown here in cyan, is actually occluded, so it can't self-assemble into an active form. It may be that this closed form is actually part of a bigger structure like a cage, as was recently published by several groups. So when NLRP3 recognizes its activation stimulus, and quite what that is still a bit of a mystery, it unfurls into this sort of extended conformation that I've shown here. And now the oligomerization domain can interact with one another in different monomers to build this sort of oligomeric wheel that we call the NLRP3 disk. So NLRP3, now we're seeing it from a side view, has now these pyrin domains in red in the middle of the disk, and that allows interactions with the inflammasome adapter ASC, who also has a pyrin domain through these pyrin-pyrin interactions. And ASC, as I mentioned, has really unusual properties. Once it binds to the inflammasome, it actually fibrillates to form these long fibers that eventually all tangle up with one another into this sort of big, gorgeous complex called the ASC effect. The ASC also has a caspase activation recruitment domain, shown here in orange. This is the card domain that interacts with the card domain of caspase 1 and therefore recruits monomers to the growing complex. And as I've mentioned, we have found that this allows for caspase 1 dimerization, for interdomain linker cleavage, and this rearranges the protease subunits to make them more stable. So caspase 1 then has activity on the inflammasome. It cleaves substrates like pro-interleukin 1 beta and pro-interleukin 18, and also this pore-forming protein called gasdermin D in grey. Now, after caspase 1 has done its job, it wants to turn itself off to prevent unwanted signaling. And so caspase 1 cleaves itself a second time, now at the card domain linker. This ejects the protease domain from the complex, and that protease domain is no longer stable. The dimers dissociate, and protease activity is terminated, turning the whole inflammasome signaling pathway off. So now we're sort of pulling back and seeing what happens with the cells. Gasdermin D here in grey is now forming pores in the plasma membrane. Actually, at the same time, IL-1 beta is lining up on the plasma membrane at the same regions, waiting to exit through these Gasdermin D pores, which also allow water in, and essentially osmotic swelling and lysis then eventually mediates pyroptotic cell death. And essentially the membrane tears, as well as the cytokines that were already released through Gasdermin D pores. You also have other alarmins being released by cells. And the sum total of all of this is to drive inflammation that drives cell recruitment to a site of infection or injury. Neutrophils and later monocytes come to the site of infection or injury, hopefully resolve the damage, clear an infection, for example, and return the system back to homeostasis, which is, of course, the primary end goal of any innate immune pathway. So that serves really as a bit of an intro to the second part of my talk, and that is how you might drug NLRP3, this problematic inflammasome sensor that senses cell stress and drives many diseases. So I was very fortunate to be recruited to this collaboration that was nucleated by Luke O'Neill in Ireland several years ago. Luke had read an old Pfizer paper that described a compound that they called CRID3 and we call MCC950. And this is actually a paper that predated the discovery of the inflammasome, and noted that this compound blocked IL-1 beta release from stimulated monocytes. And Luke, being the clever man that he is, thought, oh, I wonder if it inhibits one or other inflammasome. And this sort of developed this into a large collaboration and a lot of interest in order to pursue what exactly this CRID3 or MCC950 molecule does. So Luke collaborated with Matt Cooper, who's was at my institute, and the two of them then recruited me as an inflammasome person. And this is all work that was done by Rebecca Cole, a wonderful PhD, and continued on this same project. So basically, we investigated exactly what this molecule MCC950 does, and in this Nature Medicine paper from 2015, we showed that this molecule is a very potent and specific inhibitor of the NLRP3 inflammasome pathway and can block NLRP3 mediated diseases in mouse models, so in a mouse model of MS, as well as a mouse model of hereditary fever syndromes, as well as ex vivo in, for example, patient monocytes who have these hereditary fever syndromes driven by gain of function NLRP3 mutation. So this paper was an enormous paper and a real tour de force. But what we didn't understand at that time is precisely what MCC950 did, what the molecular target of MCC950 was, or what its mechanism of action was. And we also were very cognizant that this very potent and specific orally active NLRP3 inhibitor had enormous promise for development as a new anti-inflammatory drug. And so while we were collecting this data, we initiated a drug development program between my lab, Matt's lab, and Luke's lab. This is with Rebecca and Avril, to develop new chemical entities similar in action to MCC950 that we could commercialize and hopefully bring to the clinic. And so we patented these molecules, I think, on the same day that the paper came out, and set up a company called Inflammasome to develop this further. And Matt recently published a paper. It was a very nice review on the functions of, or the therapeutic benefit of MCC950 in different disease models. And he claims, and I believe him, that MCC950 has now shown benefit in more than 100 preclinical disease models, which is just incredible and really highlights how problematic this NLRP3 inflammasome is. Here are just three that I was involved in. A really beautiful study from Trent Woodruff showing that MCC950 blocks 3 different models of Parkinson's disease in mice and has really stunning effects. Likewise, another paper that we were involved in that shows beautiful effects of MCC950 in experimental non-alcoholic steatohepatitis, an area that we're still very interested in, and another in spontaneous colitis. So there's been a huge amount of interest in NLRP3 inhibitors and their potential for treating diseases. But the question always remained for us, what is the molecular function of MCC950? What is the molecular target and how does it actually block the inflammasome pathway specific to NLRP3? And I won't go into all the details, but 950 is an excellent tool compound. It's used widely in the field, but it's not suitable for clinical application. But we didn't know how to rationally refine this compound because we didn't have, for example, a crystal structure of the compound bound to its molecular target. What we did know is that it blocked all forms of NLRP3 activation, and there's lots of these, canonical, non-canonical, alternative, all the rest. We knew it blocked the pathway at NLRP3 or upstream of NLRP3. And actually, it was widely speculated at the time that it interacted with NEX7 to block NEX7-mediated activation of NLRP3. Again, I'm going to cut a very long story short here and just show you this really key piece of data from our medicinal chemistry magician collaborators, Avril Robertson and James Hill. They designed a photoaffinity probe of MCC950 in which they'd modified MCC950 to include a UV activatable moiety that would allow it to covalently cross-link with its molecular target, and also another moiety that was amenable to click chemistry so that we could click on biotin to pull down the compound with streptavidin. And what we found actually is that when we incubated our photoaffinity probe, macrophage lysates, added UV to chemically cross-link it to its molecular target, and then clicked on biotin and did a streptavidin pull down, we actually saw no interaction of the photoaffinity probe with NEX7 and actually a beautiful specific interaction with NLRP3. And we know that the photoaffinity probe binds to the same part of the molecule as MCC950 itself, because we can actually compete this interaction off if we add an excess of 950 before adding the UV crosslinker. So this showed us that MCC950 bound to NLRP3, but did not really indicate that it's necessarily direct. So we repeated the same experiment with recombinant NLRP3 and found exactly the same thing, showing that indeed 950 directly binds to NLRP3. So NLRP3 has this domain structure, which I showed sort of more prettily in the cartoon before. But essentially, the middle structure here, the middle domain that mediates self-oligomerization is known as the NACH domain, and it actually has ATPase activity. And we've known for a long time from the work of Jenny Ting and others that this ATPase activity of NLRP3 is really critical for its function. So NLRP3 has Walker A and Walker B motif that allows for ATP binding and hydrolysis to mediate this ATPase function, enzymatic function of the molecule. And we had reason to believe that 950 targeted the protein somewhere in this NACH domain at an area that was critical for ATPase function. And this came from data in which we did Biocore, collaborators from Griffith, Chris Day and Mike Jennings, in which we put recombinant NLRP3 on chips and flowed across ATP or MCC950. ATP binds, of course, to NLRP3 because it's an ATPase, and so does MCC950 because it's the direct interactor of NLRP3. And what was really interesting is when we added both MCC950 and ATP, we generated a really high affinity interaction between these three molecules, which suggested to us that 950 may actually be blocking the ATPase activity of NLRP3 because ATP is not able to be converted to ADP and be released from the complex. So this suggested to us that 950 might be blocking this ATPase function. And indeed, when we used a Walker B mutant of NLRP3, we saw no interaction of MCC950 with this Walker B mutant. So this suggested to us that MCC950 binds to NLRP3 in such a way to disrupt its ATPase activity. And actually, we published this back to back with a study from the Poligran lab, which came to a similar conclusion using different techniques. And actually, they were studying the structure of NLRP3 and how this structure flexes from going from the inactive form of NLRP3 through to the unwound active form, and then finally the oligomeric wheel that can signal. And both studies concluded that actually MCC950 blocks this ATPase function of NLRP3 and so blocks the development of this oligomeric wheel. And really excitingly, actually, Pablo Poligran's paper suggested that if you add 950 to preformed oligomers, you actually may be able to disassemble these oligomers, which would be very important in chronic disease. And more importantly, revert NLRP3 molecules from their open active state back into a closed and inactive form. And several papers have since come out from various groups confirming that 950 actually binds the NACH domain of NLRP3 to prevent ATPase activity. So where are we now with inflammasome inhibitors? Well, our startup progressed 2 drug candidates through successful phase 1 and phase 1b trials, and Inflammasome was recently acquired by Roche, which is extremely exciting because Roche have the capability and know-how to take this all the way to the clinic. So very excited to see what happens coming out of Roche in the next few years. Several other startups, including NodThera, IFM Tray, and Olatech, have successfully progressed NLRP3 inhibitors through phase 1 trials. Some of these also have undisclosed phase 2 data, which I'm very much looking forward to seeing. And more broadly, this story with NLRP3 has really shown that inflammasome sensors are druggable targets. And this knowledge has really fast-tracked industry interest and programs into developing new drug candidates for other inflammasomes, whether they be individual inflammasome sensors or also pan-inflammasome therapeutics like caspase-1 inhibitors, as I described in the first part of the talk. So that, of course, just leaves me to the most important of all of the slides, and that's the acknowledgements. The 2 stories I told you were led by really incredible postdocs in the lab, Dave Boucher, who's the caspase-1 guru, and Rebecca Cole, who's the MCC950 guru. These are really amazing scientists, and I'm very proud to say that they're now running their own labs at York University and Queensland University Belfast. They were very much assisted by other people in the lab, including Kai-Wen Shen, Yelena Bezbroditsa, and others in the lab. I also want to give a big shout out to, and sorry, Mercedes Monteleone, who is somewhere in here up the top there. Also a huge shout out to Matt Cooper and Avril Robertson, James Hill, who led medicinal chemistry of either the in the Inflammasome space, or Avril and James, who designed the photoaffinity probe for our MCC950 mechanism of action story. Also our collaborators at Griffith University, Mike Jennings and Chris Day, who did the FPR work, and of course Luke O'Neill for starting the whole MCC950 project. So thank you very much for your attention. I'm really happy to take any questions that you might have.
Thank you, Kate, for the wonderful talk. It was very, very interesting. We have a few questions here in the Q&A section. And the first question...
Fantastic.
Yes, indeed. So the first question comes from Brahmani Sontiana, and says, does caspase-1 has any role in autophagy other than inflammasome mechanisms? And if yes, what is the state of autophagy during inhibition of caspase-1 activation? Okay,
so the question is, does autophagy regulate caspase-1 activity? Is that right?
Yes, that would be my interpretation.
Okay. So autophagy and inflammation, as I'm sure everyone's aware here, are really intricately linked, and it's a delicate balancing act between the two. Autophagy regulates several parts of the inflammasome pathway. For example, it degrades pro-IL-1, so modifies the substrate availability. Autophagy or autophagy-mediated autosecretion has been suggested to mediate IL-1 secretion in some circumstances, although not in our standard assay systems, actually, where your serum release. I don't know of autophagy directly regulating caspase-1 activity, other than that inflammasomes are reported to be sort of essentially eaten up by autophagosomes to inhibit signaling by the pathway. I've always been a little unclear about the situations in which that might occur, because inflammasomes generally trigger cell death, and cell death is going to be much speedier than autophagy. But there are situations in which cells can survive inflammasome signaling. For example, we published in neutrophils that are infected by salmonella. These cells, although they're prone to dying, actually don't die by pyroptosis in that situation. It may be possible that in some circumstances, autophagy is actually eating up inflammasomes with active caspase, and so deactivating caspase that way, because some cells actually have they turn over caspase-1 activity much more slowly than the macrophages that turn over caspase-1 very quickly. So it's certainly possible that there are links there. I would say it's a little unclear still at the moment.
Lovely, thank you so much. That's a very clear answer. So we have another question from Ten Fazy, and the question is, are there ultrastructural data to share of the organized molecules diagrammatically shown nicely in your talk? So is there structural data available?
Yes, so there's a lot of really beautiful data coming out, and actually some of it's come out since we made our movie, and we were very pleased to see that our movie is actually reasonably accurate, which is good. So there are structures of the inactive NLRP3 molecules, which it looks like they're actually, I alluded to this in the movie, but for simplicity, I've shown them as a monomer, but they may be part of a more complex structure like a cage. And when NLRP3 is activated, it does open out. We based our structure on NLRC4, whose inactive and active structures had been solved. And it looks like NLRP3 has similar inactive and active structures. The ASC spec has been, the sort of filaments of ASC have been structurally solved by Hao Wu's group and others. Hao Wu has been doing an enormous amount of beautiful work in this area, and she's solved also the structure of the Gasdermin D pore, amongst others. Also, she's resolved caspase-1 fibrils. But I would say, for me anyway, I'm not entirely sure whether that's an in vivo, in cellulo phenomenon, or whether that's an in vitro phenomenon. So many of these structures have been solved, and they're really gorgeous structures, actually. So it's worth having a look at them, even if it's just for a bit of eye care.
Indeed, they are beautiful structures. Lovely, thank you so much. So we have another question, this time from Anthony Ravasin, and the question is, what about the natural blockers or turn-off signals of NLRP3? Can you comment on those?
That's a really interesting question. We don't know that much about natural blockers of NLRP3 signaling, or any of the inflammasomes, actually. We know about microbial evasion mechanisms, because, you know, any host pathogen interaction, there's always going to be subversion of host pathways. We don't know that much about endogenous regulation, other than to say that NLRP3 activation requires this sort of unfurling of NLRP3 from this closed structure to a more open structure. And so it's very possible that endogenous mechanisms may regulate that. There are endogenous mechanisms known, like things like post-translational modifications and subcellular location of NLRP3, that influence its signaling, but like an endogenous inhibitor or something like that is yet to be really clearly defined. We thought, the field thought several years ago, that NEK7 was going to become the universal activator of NLRP3, but it turns out that that's probably not the case in all situations. There was an idea that much like cytochrome C binds to APAF1 to mediate oligomerization of APAF1 and caspase-9 activation, something similar might happen with NEK7 and NLRP3. But Veit Hornong's data suggests that that is not probably a universal mechanism, although it is in operation in all cell types.
It's a very interesting answer and question, because I imagine that has, for many different types of mechanisms, there are natural inhibitors in the cell, and I'm sure
yes,
it's what area of research.
Yeah, well, there's actually some very old data from Jörg Chopp's lab from before I was in Jörg's lab, which suggested, for example, that heat shock proteins and SGT1 hold NLRP3 in an inactive but signal-ready state. So there may well be things like this. That's sort of a paper that people have, I think, forgotten about. You know, it may well be that there are many of these things that keep NLRP3 on a hair trigger waiting for activation. You know, they're clearly not very good inhibitors, because NLRP3 gets activated pretty easily. But I guess we'll see what happens.
Yes, indeed. Thank you. I'm sorry you have so many questions, and it's a very interesting topic, so you have to waste much of had a chance to drink some water. So another question is from Anthony Ravasin again. How similar are these to the small molecule inhibitors in mechanism of action? So I think you answered already that one, in the sense that there may be mechanisms shared between the inhibitors that you are designing and the so-called natural ways of activating the inflammasome.
Yeah, I think it's about the small molecule inhibitors. So we just wrote a review on that, actually, which so that was Pablo Pelegrin and Rebecca Cole and myself in TIPS, which goes through the mechanism of action of all of the known or published NLRP3 inhibitors. And actually, each of them work a slightly different way, but many of them target the ATPase function of NLRP3. 950, as shown by Novartis and others, acts as what they call intramolecular glue, to glue all of the domains together so that NLRP3 can't unfurl. So they sort of lock NLRP3 in this inactive state. Some of the other inhibitors that have been published block things like NEK7 NLRP3 interaction or NLRP3 ASC interaction. So there's a few different mechanisms by which these molecules can occur. but certainly blocking the ATPase function is favoured and would be a really interesting approach for other inflammasomes that involve an ATPase.
Okay, wonderful. That's a very, very clear answer. I have a small question here from Lenin Leibenberg, and it's sort of related to that. At what point does the inflammasome feel like not being active anymore?
It's a great question.
Yeah, indeed.
So we spent a lot of time wondering this question, because what was surprising to us is that in macrophages, the activity of caspase-1 is all in a burst. It all happens in about 5 minutes. And that's when all of your substrates are cleaved and the cell then is programmed to die. And so that really had us wondering, well, if everything happens so quickly, how do you get chronic disease? Is it multiple rounds of activation in new cells that then go on to die, or what? But what we've discovered over the years is that some cells and some stimuli do not provoke inflammasome-mediated cell death. And so they can live to signal a bit longer. And also some cells and some stimuli actually provoke very low level but chronic caspase-1 activity. And we played all sorts of molecular games trying to understand the determinants of this timer, and found cool things like the smaller the inflammasome, the longer the activity of caspase-1, but the weaker it was at any point. So you can actually play with the timer so that you have less activity at any one time, but chronic activity over time. So for example, if you use heterozygous cells, macrophages, so that you've got half the amount of ASK and your inflammasome is smaller, you now prolong caspase-1 activity over time. And then other cells like neutrophils that have a very small ASK spec actually have incredibly prolonged activity of caspase-1, and you now get caspase-1 dependent IL-1 production over hours rather than minutes. So there are many determinants of this timer, and I wouldn't say that we understand them all. But it's a really interesting molecular question on how this is regulated mechanistically. It
is a good question indeed. We're about to close. I mean, we have 4 minutes, so we have time for a couple of questions, if that's okay. The next question, of course, it hurts me because I'm very fond of macrophages, but it's a very interesting question that I also had. And it is, can you comment on non-hematopoietic inflammasome functions? So do they exist in other cells that are not our favorite macrophages?
Yeah, absolutely. And it hurts me too, because I'm a myeloid person. I'm a bit broader than macrophages in that I particularly like neutrophils. Yes, it is broader than the hematopoietic system. And in particular, I would say that the key cell outside of the hematopoietic system that mediates inflammasome signaling is actually epithelia. So it's your barrier surfaces, which, as you know, have a sort of pseudo-immune function in order to be able to prevent pathogen invasion. So I'm talking about gut epithelia, airway epithelia, skin epithelia. All of our barrier surfaces have inflammasomes. They may not have every inflammasome, but they have some. And particularly NLRP4 is highly expressed in gut epithelium, as is NLRP6, and also the non-canonical inflammasome. And one of the inflammasomes that's emerging as a really interesting one is actually NLRP1, which is expressed in airway epithelia and also in skin. So yeah, it's becoming an area of enormous interest to many of us. And I'm feeling like I might actually have to extend myself out of the myeloid system to other systems as well, sadly.
It's a very interesting topic. I wonder if in placenta as well, and places like that where you don't...
Well, there's actually a beautiful paper in JX Med published last year, which we were fortunate to write a preview on, which actually showed that trophoblasts have inflammasomes and drive IL-1 production to protect an embryo from infection from the mother. So yeah, they're everywhere, inflammasomes. I think we just haven't really looked hard outside of the hematopoietic system. So, so yeah, lots of really interesting questions there.
Amazing. Can I ask you a very short question for 30 seconds? Is the last question, so we don't leave anyone. It's by Christina Scherboni. And the question is, which is the influence of NLRP3 polymorphisms in the sensitivity to the inhibitor you described?
That's a great question. And there's been a bit of debate in that in the literature. So in our hands, all of the inhibitors that all of the polymorphisms, particularly the disease-associated polymorphisms that we have tested are inhibited by MCC950. What I suspect is the case is that the dose-response relationship mutations, particularly the mutations that are in the NUC domain around the ATPase domains. So we tend to use MCC950 at a very high concentration that'll just hit everything. But what I suspect is that as you change particularly key residues in the ATPase domain, so around 300, give or take 50 residues, that you may need to have a higher dose of MCC950 in order to block inflammasome signaling. But our inflammasome drug candidate were tested, one of them was tested in a CAPS patient where it showed efficacy. So, that's one bit of human data. We've also got several pieces of data in the 2015 Nature Medicine paper, which showed that 4 different mutations, gain-of-function mutations of NLRP3 are still blocked by MCC950 in human patient monocytes ex vivo. So, yeah, it's that's probably all I can say at the moment. But industry would probably know the answer better than I about that. But I am yet to see data that really convinces me that 950 will not work in patient-associated mutations. You may just need to increase the dose.
Okay, thank you so much, Kate. That's very clear. We have reached 10 a.m. So I wanted to thank you so much for the wonderful seminar. And I'm sure all those that could attend, and we had over 100 people, or close to 100 people attending, had this wonderful pleasure, as I did. So thank you very much for your time and your expertise. And we wanted to leave you with this notification for the 18th International Congress of Immunology that is going to take place in South Africa from the 27th of November until the 22nd of December of 2023. So keep an eye on this wonderful opportunity to meet colleagues of all over the world. So if there is nothing else, I thank you all for your participation today, and especially Professor Kay Schroder, who really opened our eyes about the inflammasomes in the body. So thanks, everyone.